Good morning, everyone. Welcome back. I'm Matthias Balloff. I will be chairing the first session. I was asked by our chairman to remind all speakers that they should stick to the schedule. So we had yesterday wonderful discussions, endless discussions. I think we have to cut it down a little bit. Okay, the first speaker is Phan Anh Tuan. I hope I didn't butcher the name. With the title, where's your okay, here we go. The structure dynamics and recognition of G-quadruplex nucleic acids. Please.
Good morning, everyone. Thank you for coming such early session. Thanks organizers for inviting me to present our work. I tried to, through 30 minutes, I hope, to give. Oh, okay, 29. To present our work on structure, dynamic, and recognition of G-quadruplex nucleic acid. Most of the illustration are from our work in the past 10 years. So it's a title. The talk will be about the structures, the dynamics, and the recognition of G-quadruplex. For interest of the audience who might not be familiar with this kind of nucleic acid structure, I will have a section to introduce to you the G-quadruplex topologies. So this is simplistic schematic of G-quadruplex. When you have a lot of guanine, they can come together in planar arrangement like this, and many layers, we call tetrad, can stack on each other, stabilized by cation, to form a very stable structure. In this schematic, I don't show any strand orientation or connection. It just simply the score of the structures. This is some of the structure from our group. You can see a zoo of variety of different possible structures formed by just a piece of DNA from different places in the human genome, the telomere, the promoters, or some artificial DNA. It could be a single strand of DNA fold back and forth into a structure. It could be assembly of two or more molecules together. The quadruplex structure also polymorphic in the sense that even the same sequence from the human telomere, TTA, GGG, can adopt variety of structures depending on the condition, environment you put them in. So in this, you see they're different, but in which sense? So in the sense, for example, the orientation of strand 5', 3' of DNA going in different orientation, the way that you connect the strand by a loop, we call loop in red here, or here you can see the color coded of the base. Here what I meant is that for a nucleotide, it can adopt two different orientation called syn and anti. It simply changes orientation of the base with respect to the sugar. So from our group, we try to see that diversity from the point of view of elements of the structure. So for example, if here's a quadruplex block, here I represent one layer of tetrad by one shape here. You can connect the two corners, we call loops. So there are different ways of connecting the corner so that result in different type of loops. And if you take the core, they have 4 strands, and like double helix, there are 2 strands. So if 4 strands, there are how many ways that 4 strands can be oriented with each other? There are 4 different ways. Either they all in the same way, or called parallel, or the 3 in one direction, 1 opposite, or 2 up, 2 down in different manner across the diagonal or across the side. So that's all possible way that 4 strands can orient with each other. However, if you look at this schematically, you might not see the big difference between different topology. Let us look into the geometry of such structure a little bit. Here's a top view of a quadruplex. Here 4G. The distance are measured in angstrom, depending which atom to which atom I point. If I 4 strands go from the board toward us, we have 4 grooves of DNA. In this case, they are of medium size here, like 14 or 16 angstrom, depending which atom you measure. Now, and then all the nucleotides are in anti-conformation. You remember orientation between the base and the sugar. Now let me flip one of the strand into the board, because you know there's a different way of orienting the strand. Okay, just by geometry, this has to be flipped in syn, because otherwise it would break the bond, right? And then you see appearing a wide group and a narrow group. If I do another exercise with the other strand, you have another. I can do another one. So I hope that I could illustrate to you that actually 4 different orientation of strand result in very different geometric arrangement of the groove size and so on. Okay, so that's just the introduction. So let's get back to our topic. First about the structures. To illustrate one of the key word about quadruplex, probably about the polymorphism, meaning that different structure can be formed, and one of the best example, or most studied, is about the telomere, or the end of chromosome. So here's a schematic representation of the human telomere end, where you have a single strand protrusion in the 3' end. And it was shown that for most of the cancer cells, telomerase most of the cancer cells would require telomerase to elongate the telomere using the substrate, which is the single strand of the DNA. However, if quadruplex can be formed, then that could inhibit the telomerase. So that's for the research of many different groups. And the question we ask is, what's the structure of this sequence? So about over 25 years ago, many groups study quadruplex structures. I think even some pioneers sitting in the audience here, Daniela wrote, has a structure of different species. And a model of the quadruplex structure at that time is 4 repeats, which is a minimal repeat to fold back and forth into a structure. And this 25 years ago structure of NMR in sodium solution showed that the piece of human telomere can fold back and forth into this fold. The field was quiet for about 10 years until another paper appeared showing a very beautiful structure, but in potassium crystal. So the same piece of DNA, but crystallized in completely different shape. It looked beautiful, and it's appeared in Nature, so everybody come in and study and ask the question, so what is the relevant structure? Because it could be different fold. Also, it's important because in the cell you have more potassium, so potassium seems to be more relevant. But yet the cell may be more close to solution conditions, so maybe solution is more important than crystal. Just to cut the long story short, about hundreds of papers, even up to now, talking about what the structure in potassium solution. We could show that at least a piece of DNA like that can fold in very different fold. I won't go into the detail, just to say that they're actually different. They can also form a fold that don't have all 3 layers of tetrad, but only 2 layers. And to summarize that, same piece of DNA or similar piece of DNA in different environment, sodium, potassium, can form very different structure, including a mixed tetrad. So so it's fun. The polymer can adopt different environment, different conformation, depending how you have the environment, or even the way that you fold the molecule. Yesterday, in the talk also, there was mentioned, maybe also important question, what about the crowding environment? Again, that can be mimicked in a test tube by adding co-solute like PEG. So if you use circular dichroism, this piece of DNA, it folds this structure, giving that blue CD signature. You add PEG in, you see this signature. Very big change in the signature. And of course, you can look at NMR, you see also a change. You can do it with different forms that you saw earlier. In all cases, you see the same changes. You can use also anything you find in the lab, not just PEG 200, but also PEG with bigger molecular weight like 8,000, 35,000, even acetone nitride, ethanol, even DMSO, or whatever we found in the lab, which is put in, and even polysaccharide. So it's not about just hydrophobic or something. In most cases, we see very similar transition. You can monitor that transition with the time. For example, you can look at the CD and look at this peak or this peak. You can see that it can form in just a few minutes if the temperature is high, or a few hours if it's low. You can unfold again if you put back into more dilute environment. So as you do have NMR peaks, so we can assign the structures, solve the structures, and it turned out to be Barland structures like the crystal structures. So people who solved the crystal structure was very happy because they were attacked for many years that it's not relevant. Let's try to understand actually what it is behind. So we try to do some simulation. We put the molecule in a box containing water and mixture with something else, simple for simulation, which is ethanol. And here's the structure that is Barland, and here other form. In red, you see the plot for the box pixel that occupied by ethanol. You can see that maybe it doesn't explain, but it helps us to understand that why this one is more favored when you deplete water. When you deplete water, this fold tend to be more favored. And then we look back at crystal structure. It turned out that when they crystallize, they also use the reagent that deplete water. As well as you look at the water content in the crystal, there's about only 60%. So in the crystal, you can even say that macromolecules are crowding agents themselves, and they deplete water. So I think the summaries of this work can be that thermodynamically, anything can be converted to this fold. But of course, in the cell, it's still open question because it's also the kinetic. It takes minutes to hours to be converted to this fold that you've seen earlier. And also, there are other components like proteins. Okay, so I hope that through telomere, which is a well-studied object, you see that the quadruplex is polymorphic. But let's just very quickly go through some other quadruplex forming motifs. So typically, people look at that motif intramolecular that contain this motif. So the 4 track of G required for folding back and forth 4 times the piece of DNA. If you look at the stability as a function of the length of one of the loops, you can see that the longer the loop, the less stable the structure. So arbitrarily, people stop at 7. Even with that limitation, look through human genome, you have 300,000 possible places where such motif exists. So I would show in a few next slides that actually that motif, it's just a basic motif, but the space could be even broader. For example, in some promoters, you can see that the fold looks like very twisted. Or in this case, you also have very twisted fold. In some mini satellites, you have very big loop, or you can have also very twisted structures. You also can have structure with small bunch, bigger bunch, 1, 2, 3 bunches, bunch in different places, or bunches in different folds, meaning that the G track can be interrupted with many different things, and still you have enough G, you still can fold into the structures. You can also have the structure not having all the G, but missing some G. So all of this relax significantly the requirement for the sequence, so the space should be much bigger. I will come back to that issue in some of the subset of structure that we saw. And surprisingly, recently we accidentally fall into a crystal and NMR structure of a sequence that is left-handed manner. So most of the quadruplex studies so far go in the right-handed manner. If you know about left hand and right hand, duplex DNA B and Z form, you would know that mostly on the B form, you have the way of stacking from 5' to 3' in this direction, which is about plus 30 degrees. Left-handed, like in some of the steps of the Z DNA, would go in the reverse, minus 25 in this case. And in the CD, you will see the signature nearly mirror image, because one is the right hand, one is the left hand. We yet to fully understand the basis for for this structure. But of course, this must be in the configuration space that allow that you already seen in the zDNA. And what we talk about so far is individual unit of quadruplex. But like you heard yesterday in the telomere, usually you have tandem of the sequence in the telomere, or in some mini satellite, also tandem of G sequence repeats many times. We could show that it could form some sort of structure called bit on a string or bear necklace type of structure where you have quadruplex connected with single strand. I will spend about 2-3 slides talking about another phenomenon. When you have that might be relevant to formation, like in bacterial matrix. When you have pieces of G-reach, whether they can self-assemble into structure quadruplex base. So we study this piece of DNA, just 10 nucleotides. That had been studied for a long time that this piece of DNA could self-assemble into a structure called wire, called G-wire. So in this work, we try to look again back at that structure, but at high resolution and in aqueous solution, which actually allow us to go to high resolution. You can see a few wires here in coexistence with reference duplexes. Here's a plot of height. So you see duplex and quadruplex. Because we could have high resolution down to nanometer, you start to see periodicity in the wire. You have two types, one about 4.3, the other one 2.2. So looking at this structure, would you see that it's a left hand or right hand structures? What do you think? Of course, in our structure, we have all the structure in mixture. Some orient this way, the other way, right? But now I orient them like this. Would you see that left hand or right hand? I hear somebody say left already, right? Let's say I have a helix. If I make a right hand, the incline should be on the right, no matter how I turn my molecule. If it's left hand, the incline should be on the left. So clearly this structure is left hand. Of course, if we look at the CD, there's still a right hand. I will try to explain to you why we would see that. Okay, so again, here's we see some left hand pattern, some zigzag pattern, and we try to rebuild the model, assembling of different pieces of DNA. And you can see that this is a right hand progression of the structure. However, you have a feature that going in the left hand direction. Okay, that feature actually is the bunch in the structure. So sometimes you see the feature, but it's not about progression of the backbone, which is canonical for pairwise nucleotide, but it's about some feature that protrude to the left hand. Okay, we talk a lot about quadruplex. Then can it coexist with the duplex, or how compatible it is with the duplex? So to examine that question, let's look back again the duplex. If you look at the dimension in geometry, phosphate distance about 18 angstrom on a minor group. Quadruplex has 4 groups. Okay, so let's try to see whether they can be connected. So to do that, we try to connect a hairpin duplex to the quadruplex. We show that actually there are at least 5 different ways to connect a duplex to a quadruplex, either adjacent or sideways. I just give you 2 examples here. One, the connection between minor group with the group that you saw in some earlier slide. You see the geometry are very close, so they seamlessly can be connected, like you can see here. You can also see quadruplex and duplex can connect sideways. In this case, the simple solution is to flip out 2 bases. That you have seen in the BZ connection crystal structure. So the nature found very simple solution connecting from right hand to the left hand by just flipping out 1 base. So here also connecting 2 helicity by flipping 1 base. So since you can connect 1 in different way, you can connect many like this. You can see feature in NMR. If you look at the sequence, they don't resemble to quadruplex forming sequence. You can have a structure. And then you can also connect multiple duplex hairpin on the same loop. You can look at this in a reverse way, like quadruplex. It's like four-way junction and put a quadruplex in one of the arm. How about the stability? So you can see here, example, using NMR, you can monitor peak of a quadruplex. And in more base pair grow, you can see duplex forming with longer stem. And if you measure the melting temperature, you probably remember that for unstructured loop, the longer the loop, the less stable, the lower the TM. But for the stem loop, you don't decrease, but slightly increase the stability. So such structure is feasible. You can form by G-track very distant in the genomic sequence. Okay, so I will very briefly talk about dynamics. So you already have seen that from one form of the quadruplex can go to the other form in a matter of seconds, minutes, or hours. Inside the structures, it's relatively stable. The tetrad, unlike the duplex, you have a breathing of base pair happen millisecond time scale. But in the quadruplex, sometimes if you measure exchange of this proton, it stays by extrapolation at room temperature to 10 years over. But actually, there's some part of the structure is still dynamic. For example, these groups, these amino groups. You can see that in NMR, you can see that if you have the group that rotates slowly, you have two distinct conformations. When they rotate very, very fast at higher temperature, it averages out in the middle. You can see that example in some case, for example, here it is broad, it rotates fast. If you put a methyl, you freeze out the rotation, it's sharp. Or you can freeze the rotation by hydrogen bond. You can freeze the rotation by ligand binding. Just to give some flavor that actually the molecule have some dynamic feature inside it, although the core is pretty rigid. For the sake of time, I'm running a bit fast. I'll go to the last part of my talk. Okay, so you have such structures. If it exists in vivo or it is the object, artificial object for application, how that structure can be recognized out of other type of nucleic acid. First, about small molecules. So chemists have screened many thousands of compounds, and some of them have been found to be very potent in stabilizing quadruplex or having biological activity. So here, one of them is a derivative of a compound called telomere. We saw the structure when we saw that the feature of recognition of this compound is by stacking on the end and provide electrostatic interaction to the phosphate backbone. Another potent compound, again, shows similar feature with positive charge here close to the phosphate. You can even see the individual moiety inside the compound by simulation could be in movement together with some bases. How about the proteins? So we were lucky to fall into a helicase that was shown to be specific to quadruplex using this particular conserved motif called RSM. We could show that this structure is about 100-fold more specific to one type of quadruplex. There are many azure quadruplex, but you have one type called parallel quadruplex, and it has high affinity to that type. And the structure shows that this piece of protein or peptide actually sits on top of the quadruplex, similar to the compound ligand that we saw selected to the quadruplex. And if you look at the detail, you see that it uses hydrophobic residue to dock on the platform of G4. It also used 3 positively charged residue, like 3 fingers, but positioning correctly into 3 different groups, with specific dimension of quadruplex, right, to have contact with phosphate backbone. So again, you see that actually protein use the same strategy that small molecule use, using the platform of G tetrad and electrostatic interaction. If you think about that, actually, what distinguishes quadruplex from all the other forms of the nucleic acid? They all have negative phosphate, but one particular feature is that platform that can be used for stacking, either cell stacking or stack of ligand or protein recognition, because that platform it's quite unique geometry and hydrophobicity in terms of feature. Okay, so one question is, okay, so we have over 300,000 possible places for quadruplex. There's interest to specifically target one quadruplex or few quadruplex out of the rest. So we propose that you can find a place where you have both quadruplex and duplex coexist. That could be found either in at the telomere, transcription site, replication, and so on. And we propose that actually we can bind very specifically to the duplex, but high affinity and specific to the quadruplex. That's how you can distinguish from one quadruplex to the other. And together with the Sugiyama lab in Kyoto University, he synthesized a compound that contains polyamide that does bind both components of the structure. Okay, so I ran a bit faster then, so let me conclude my talk. I hope now you are convinced that quadruplex structure is polymorphic, different shape, different forms, different structure elements. They can be connected with a duplex, compatible with duplex. Even though it's quite rigid, but some structure elements, dynamic, and it's also interconvert between forms. And the exposed state chart and a backbone quadruplex can be specifically recognized by ligand, both small molecules and protein. So let me thank my group members, Bas and the Current, and my collaborator and funding agency, and thank you for your attention.
And thank you for leaving a couple of minutes of our time. Questions, please. Please, Rudi.
Many times you showed the potassium ion with your structures.
Yes.
I would like to know what's the state of this ion. Is it mobile inside the quadruplex? Is it just absorbed somewhere and fixed? What are the details?
Actually, that's a good question. It is hard to study directly the potassium. So in the crystal structure, we see the ion like potassium sit in between, or we see the electron density between the two layers. In NMR, we cannot observe potassium directly, so people observe ammonium ion. And depending on the structure, even though it looks the same, but sometimes it could be slightly tighter, we do observe the movement in order of millisecond time.
Inside
the channel, exchange between one side to the other in millisecond time scale.
So that should change the electrostatic interactions between these quadruplexes, no?
You mean during the movement?
Yeah, if it's mobile inside, that means something.
But that is millisecond time scale. So it probably jump from one side and the next one jump in. Yeah. We also don't know whether ion H3O plus can also occupy, but the size seems to be small, and experimentally we have not detected.
Okay, thank you.
You talked about these crowders in the beginning, and there are many types. But what about the interactions between the crowders and DNA? I mean, polyethylene glycol is quite hydrophobic, so it's the hydrophobic interactions will probably play a role. It's just not just an inert thing.
Yes, you're right. So actually, it might not PEG might not be innocent because it also can have some interaction favorably for one structure over the other. However, the effect has been seen for all sorts of ligands, including polysaccharide, even a little bit in smaller extent in BSA, just the protein, ethanol, DMSO. I think there's some fraction of the effect as well as the effect of losing water, of depletion of water. They both contribute to the inversion.
As well, I was really intrigued by the kinetics you showed. Such a small object needs hours to change the conformation. How come?
I think that depends on the barrier energy between the folded state and unfolded state. And I think the key thing, comes from the stacking part. Mm-hmm. So some examples that we show mainly contain 3 layers. But we do study some object contain 6 layers. So we put in boiling water. Mm-hmm. We boil it for 10 minutes. It doesn't dissociate. The reason we can observe that is the solution is D2O. If it ever open, we're supposed to see the peak disappear. We boil it 10 minutes, the peaks of immunoproton is there. That means the molecule intact like a rope for 6 layers. And I think the main driving is the stacking. It doesn't want to release out.
And you see Arrhenius type kinetics if you change the if you change the temperature.
Yes.
It's a huge activation energy, obviously.
Yes,
Okay.
yes. I have a weak yes because it had little study, but
Okay.
yes, in some of the few cases we do see that behavior.
So I can't see any more questions. Let's thank our speaker once more.